Aloha. The University of Hawaii at Hilo Tropical Conservation Biology and Environmental Science would like to welcome you today to listen to Jeremy Schrader's presentation. This is part of the requirements for his Masters of Science. Today, we have the honor of Dr. Jolene Sutton to introduce Jeremy. Jolene.
Thank you, Lisa. So thank you everybody for joining us today. It really is my privilege to be able to introduce Jeremy Schrader for this component in his Masters of Tropical Conservation, Biology, and Environmental Science program. So many of you already know Jeremy. He comes to us today from his home in California, and unfortunately we're not all together, but hopefully one day soon. Jeremy earned his Bachelor's of Science in California at the University of California, San Diego in 2015. And I first met Jeremy in November of 2016 when he first reached out to me, sent me an email, sent me his CV and a letter to say that he was interested in joining our graduate program and also my research group. And, you know, I really remember that first meeting well. Jeremy, I don't know if you do.
That's good. I'm glad to hear that. I do too, no, definitely.
I remember it very well. And particularly what I remember is, for those of you that know Jeremy, his boundless enthusiasm and energy. It struck me right away. During that first meeting, he told me how passionate he was about teaching and educating in biology and biodiversity, and his interest in doing applied research that would be important for conservation and biodiversity management. And those really, you know, from what I've learned of Jeremy for the last few years, those are his two great passions in science, are both education and doing something that he feels is really important and which really matters in conservation. And then, you know, the last couple of years being able to work alongside Jeremy has been really fun as well, especially I've had the privilege to work with him in teaching conservation genetics in the undergraduate genetics lab. And I really will say that, Jeremy, you have such a great strength as an educator, and students really respond to you really well and to your passion. And so, you know, keep at that for sure. The other thing that Jeremy's been doing, of course, he's not just been teaching, he's been doing a lot of that, and he's been doing it really well, but he's also been doing really important research. And so he's going to tell you a little bit more about that today. But one of the things that he's been able to accomplish over these last couple of years is to develop a simple tool. It wasn't simple to develop, but it's simple to use for managers to be able to apply in conservation breeding programs. And he focused on the ʻalalā for his research. But this tool would actually apply to other conservation breeding programs as well for managed species in captivity. So with that, I'm going to let Jeremy take it away. If we were all in the same room, I would say please join me in a round of applause, but perhaps we'll just do a virtual applause today. But glad to have you here, Jeremy. Take it away.
Thank you very much for that very kind introduction, Jolene. I very much appreciate it. So, hello, everybody. As has been mentioned, my name is Jeremy Schrader. I'm very pleased to share with you today my internship presentation entitled In Aid of the ʻalalā, a Multifaceted Internship in Applied Conservation. Jolene has given us a brief little taste of what's to come, but I hope to, of course, explain in more detail about this tool that I developed and also some of the other work that I was fortunate enough to be able to participate in during my internship. Before we get started in earnest, I just have a quick disclosure. So because the species I'll be talking about are highly endangered, the sensitive data about them isn't in the public domain. So the graphs and the other materials I'll be showing you today are simulations of real data intended for this presentation only. So they represent the real trends, but I've just made them for this presentation as materials for this only. So, all right, with that said, thank you very much for attending and being here with me in this, this format that we have, and let's get started. So I definitely want to start by acknowledging all the wonderful people and organizations that allowed me to undertake this work. So that includes the UH Hilo TCBES Internship Track Coordinator Lisa Canale, my wonderful mentors, Jolene Sutton and Bryce Masuda. Also some specific Hilo personnel, Rebecca Ostertag, Tracy Wiegner, Shelby Wong, among many others, and also some personnel at KBCC, Amy Kuhar and Allison Flanagan. So without these people and many more than I could possibly list on the slide, this would not be possible. I also, of course, have some logos here at the bottom of the partners. So my sincerest thanks to everyone who helped me along the way. And with that, let's jump into the topic. So first, a brief background on Hawaii's avifauna. So there are over 300 species in the Hawaiian archipelago. Of those, about 64 are endemic, which means they're found only in the archipelago or oftentimes on only certain islands. So of those 64 endemic species, unfortunately, about 50% are extinct. And of the remaining 50%, more than half of those are endangered at some level. So clearly there is a need for conservation action. I have some pictures of some example species. So clockwise from the top left, we have the eevi. We have covered up, I do at least, the kiwikiu in the top right, the palila, the pueohi, the nene, and at center, the ʻalalā, which, as Jolene mentioned, was the focus of much of my work. So I'll be talking a little bit more about them in a minute here. But why do we have such a high proportion of endangered species? Well, there's been a slew of historic and ongoing threats to these birds. One of the main ones being disease. Some of you may be familiar with avian malaria and the difficulties that it's been causing. These birds evolved on an island without mosquitoes, so they have no natural resistance to or very little natural resistance to malaria, and therefore it's very devastating, or can be, to them. Another is habitat loss, which of course is an ongoing problem for much of the world's biodiversity, but is especially impactful on these species, many of which have limited ranges to begin with. And also exotic species. So other species from somewhere else coming in and potentially competing for the same resources. So one possible example would be the Japanese white-eye, an introduced species, eats some of the same fruits that these birds might. So they may be putting additional pressure on them. So with that background, I want to talk also about my wonderful partner, the Keauhou Bird Conservation Center, which I'll be referring to as KBCC for short. So KBCC supports the Hawaii Endangered Bird Conservation Program, which is funded through the San Diego Zoo Global's Institute for Conservation Research. And KBCC is located in Volcano, Hawaii, which is where I did most of my work. So in addition, it's partnered with U.S. Fish and Wildlife Service and the State of Hawaii Division of Forestry and Wildlife, which helps to accomplish these conservation goals that have been set. And it also works in tandem with its essentially sister site, the Maui Bird Conservation Center in Olinda, Maui. So together, these facilities house the entire population of living ʻalalā. So that'll be a point I come back to frequently. But in essence, they are the backbone of the conservation program for this bird and do considerable work for many others as well. So these conservation and research efforts include, of course, as I just mentioned, the ʻalalā, but they also include some of these other birds I've mentioned, such as the kiwikiu, the palila, pueohi, akakiki, and the akakee. And many of these birds are actually, they're all critically endangered aside from the ʻalalā, which is actually extinct in the wild. So these are definitely in need of the resources and the support that we can provide them to steward the species in this kind of changing and difficult world we find ourselves in. And additionally, besides doing work with these birds, there's also projects including reforestation efforts on site at KBCC and outreach initiatives at events such as the Hawaii Island Festival of Birds, getting the word out about the needs of these species for our support to bring them back from the brink, so to speak, and to, of course, celebrate their historic place in the biodiversity and the culture of Hawaii as well. So I'm going to be focusing mostly on the ʻalalā, so I'd like to give you some more detailed background. first off, the ʻalalā is historically has served as a seed disperser across the island. It consumes native fruits such as pukeave, kolea launui, and olapa, among others. And this is especially important because the ʻalalā, being of a larger size and a larger gape, being able to consume a wider variety of fruits than some of the other species, it can disperse those seeds through the forest and plays an important role in the ecology of these forests. In addition to its importance in that realm, it's also got cultural ties to the Native Hawaiian ancestor spirits and is mentioned in the Olelo No'eau, which is the collection of historical Native Hawaiian wisdom and proverbs. So it has an important role to play in several different arenas. And now on to the not so good part. It's currently extinct in the wild, so all birds living are currently in captivity with less than 200 total individuals. Efforts at reintroducing them are ongoing. And that means that all the ones in captivity, that entire population is housed and bred at KBCC and MBCC. Excuse me. And one important detail to note that I'll be coming back to as well is that all living members of the population are descended from 9 original founders. So 9 birds caught from the wild before their extirpation in 2002. Those birds' lineages have essentially, they have given birth to all of the ʻalalā we have today. So that's going to play a greater role later on in my talk, but for now, that's some background for you. Okay, so I've shared with you a little bit about where I worked and the species that I worked on. So you may be wondering at this point, what did I do? So first off, like many projects, this started with a data review. And the goal was to unify the format of data that we've been gathering about these captive ʻalalā and other species as well. So as you can imagine, these birds are closely monitored in captivity. There are many, many different data points captured about them, not only fully matured or juvenile birds, but also each egg that is laid. So as you can imagine, this results in many, many data points being gathered. And this is the way that these have been kept has changed throughout the years. So as you can imagine, we have some paper records. We have some different computer records and from different eras, so to speak. And also more recently, things like database records stored on the cloud, accessible to everybody. So all of these, when we first set out to do this project, they were all kind of a little bit different from each other. So the first step was to clean and kind of polish them up into a nice consolidated best available data set. So this I undertook together with my collaborators, Jolene Sutton and Allison Flanagan. We took these data sets and tried to consolidate the different sources they came from into one unified format, merge the different terms that might be referring to the same thing, for instance, and also, of course, double check for errors and try and go back to the hard copy and double check along the way to make sure that we truly did have the best representation of the data that we could. So this not only helped establish a baseline for the work that I'm about to tell you about, but also for further studies in the ʻalalā, and also in this process, we reviewed data pertaining to many of the other bird species as well. So knowing that we had our best available data set ready to go, my work that I chose to focus on was developing this tool that has been mentioned, which I've christened the Founder Lineage Integrated Calculator, or FLIC for short. And the goal of this tool was to help preserve genetic diversity and reduce inbreeding over time in the captive ʻalalā. So some of you may be familiar with what this means, but I know some of us may need a little bit of help. So I'm going to be talking about that. First off, I would like to say that genetic diversity, there's many potential ways to measure it. Today, I'm going to be focusing on 2 forms, allelic diversity and heterozygosity, which I'm going to be talking about more in a minute here. But I'm going to be focusing on those 2 forms, but just know there are many other ways you could measure genetic diversity. And in addition to having this tool help preserve those forms, I also wanted to use it to be able to help managers reduce inbreeding over time in the captive ʻalalā. So I'm going to focus on the inbreeding first. why would we want to reduce this inbreeding or mating between close relatives? Well, in many species, or in several, inbreeding is associated with lower survivorship and decreased heterozygosity, which, as I just mentioned, is one potential measure of genetic diversity. So anytime that you have fewer individuals being able to survive and a potential decrease in the amount of genetic diversity they have, you can see a problem in the future looming when you see those impacting this already very small and vulnerable population. So I won't make you just take my word for it, of course. I will present you this graph that basically details how inbreeding affects survivorship across the life stages of the ʻalalā. So in this graph, you can see I have a paired bar plot with a blue bar on the left half and a green bar on the right half of each life stage, representing in blue the inbred birds and in green the outbred birds. Along the x-axis, you'll see there's 4 stages. We have non-viable eggs, followed by death before hatching, and then death before maturity and recruitment, which means successfully entering the population and having offspring of your own. So on the y-axis, we have the number of individuals. As you can imagine, it goes from low to high. And you can see in the first 3 stages, the number of non-viable eggs, the number of deaths before hatching, and the number of deaths before maturity. We see that the inbred population of birds had more deaths. So this, of course, means fewer of them are living. And this is reflected, of course, in the last stage of recruitment. You can see that more outbred birds successfully survived until recruitment, whereas fewer inbred birds did. So this is, of course, a trend that you want to minimize as much as possible if inbreeding is causing them to not survive as long as we would like. Now, also, as I mentioned, inbreeding is associated with decreased heterozygosity, which can limit future adaptability. So I have depicted here an imaginary pedigree, 2 lineages represented in orange and green. And then on the bottom here, 2 offspring, one from an undesirable pairing in red and one from a more desirable pairing in blue. So you can see at left of the pedigree, we have a double black line representing mating between close siblings, close family members, in this case, full siblings, producing this offspring in red. So when this occurs, when inbreeding occurs, it can have negative effects, not only on the ability of the individual to survive, but also over time, it could reduce genetic diversity in the form of heterozygosity in the population. So these are 2 impacts we certainly want to avoid as much as possible. And this brings me to the metric that I decided to focus on for this tool, for FLIC, and that is founder representation. So what is founder representation? Well, it's the proportion of total genetic diversity associated with each founder and its offspring. And as some of you may remember, as I mentioned earlier, there are only 9 individual founders that make up the genetic heritage of all of the living ʻalalā today. So this is important because focusing on founder representation gives us 2 avenues to address the problems I was just talking about with inbreeding. If a lineage is lost, its alleles are as well. So by focusing on founder representation, making sure that each lineage survives and has some representation, we avoid a total loss of allelic diversity from that lineage. So as you can imagine with this picture here at right, you have 3 imaginary lineages and one of them is wiped out. The allelic diversity that came with that lineage is also gone. And since allelic diversity only accrues slowly over time, generally, due to genetic mutation, once these alleles are gone, they're essentially gone for good. There's not really a way to get them back. And this could potentially cause problems down the line, if not right away. Now, founder representation, focusing on founder representation also allows us to examine whether we have over or underrepresented lineages. And this, when we have this situation, this spread of lineages that are not equally represented, this can make not only avoiding inbreeding more difficult, but it can also further increase the problem of having decreased heterozygosity. So explaining that a little bit more, again, I have 3 imaginary lineages here in red, blue, and green. you can see that here I've represented them with equal numbers of offspring. So we have a group of circles and squares for each that's equivalent. But if we have an unequal representation amongst those lineages, you can imagine that not only do these individuals outnumber in the red lineage, outnumber the blue and green, making it more difficult to find mates for them that are not of their own lineage, but it also can mean that heterozygosity is decreasing as the alleles belonging to the red lineage become more and more prevalent in the population. So by using the lens of founder representation for this tool, we can try and address both the skewed representation of alleles within the population as a whole by equalizing lineages, and also make sure we're keeping track of how well represented each one is so we don't lose one entirely and the alleles that go with it. So with all of that background said, what does this look like? So basically, FLIC is intended to track the representation of each founder's lineage over time, and it spits out a graph such as this. So you can see along the x-axis, I have a span of years, which is intended to be flexible depending on whoever is running the program. They can select a period. I've just chosen to represent from before 1980. And along the y-axis, we can see the founder proportion. So that would be the percentage out of 100% that each lineage occupies of the total genetic diversity. So given that we have 9 individual founders, and they sum to 100%, if you divide that, an equal representation would be about 11%. So by looking at this graph, you can see that this can help us determine which lineages are over or underrepresented, either above this yellow arrow, which approximates 11% on the graph, or below this yellow arrow, which would be underrepresented. So by enabling managers to have access to this additional tool, of course, this is in addition to many other plans and steps and ongoing efforts to help conserve the species, this adds one additional tool to that box. So managers can analyze the trends they see. They can select a specific span of years, specific set of lineages that they want to look at. And using this tool and the flexible parameters, ideally, they can use this as an additional item to analyze and make an informed decision about where they want to take the population moving forward. So that focus on founder representation, just to tie it all together, as I mentioned, is key, given that it can allow us to focus on lineages such as you may see at the bottom right here, this one in green that's approaching the 0 line. That one is in danger of being lost. So we would see a loss of allelic diversity, which we certainly don't want. But also, as I mentioned, monitoring which lineages may be over or underrepresented, which may be skewing the alleles in the population and changing the heterozygosity, which is also critical to maintaining genetic diversity in the species. So those 2 avenues can be provided for using the same metric and in this tool. All right, so talked a lot about the fun computer stuff that I did, but of course there was also many other exciting opportunities I got to participate in. So one of these things was the operational support that I provided, and one of the main components of which was preparing bird diets. So you can see it right, a picture of the example ʻalalā diet. There was a variety of different tailored diets for each species. I worked for about 5 hours each day on preparing 2 days worth of meals for over 100 ʻalalā, about 30 plus palila and 30 plus akikiki and akike'e, and sometimes with some small changes or additions made to that. So quite a considerable number of trays of food, as you can see in this picture. And it was very important to maintain a consistent standard. So these birds, given how small their population, we want to take good care of them. So making sure each diet meets a high standard and sometimes even being individualized on a bird by bird basis was crucial. And I was very happy to be able to support, of course, this very important element underlying the captive breeding program of the ʻalalā and the other birds. Now, not only did I get to work on the diets, but I also provided support for some of the other operational components, such as the reforestation projects and native plant seedlings that are constantly being replenished from the stock at KBCC. So I would take I would take seeds from these different native plants. You can see in the picture right, I have an example from a koa tree seedling bed that I planted. I would take these plant seeds, extract them, sow them into beds, give them the appropriate growth medium and so forth, and give them a nice little habitat to grow up in and join the slew of greenhouse warriors ready to reforest up in KBCC. So that was an exciting opportunity as well. And beyond this, in those other hours after I finished the diets, I also did many other odd jobs, so to speak. So this included everything from aviary upkeep to producing new enrichment items. And something you should know about the ʻalalā is they're very intelligent. They actually, as you can see in this picture here, may sometimes use tools. And they have a very keen mind. So that means they like to investigate anything new in their enclosure and oftentimes mess with it and destroy it in whatever way they can. So they constantly need new, fun, exciting items to keep their attention and keep them happy. So that was a real pleasure to be able to participate in those as well. Now, beyond the work that I undertook for my internship at KBCC, I also, of course, took some classes and, as Jolene mentioned, had the very wonderful opportunity to participate as an instructor in some courses too. So what better place, of course, can you imagine to do field and lab courses than Hawaii? Many of them, we were lucky enough to be able to include field components or work on organisms directly from the surrounding area. So for instance, Jolene and I, for our genetics course, had the opportunity to examine samples of oysters and determine their species to see where they came from and what species they were. This also included many other things like going out in the field with students to do individual projects in the forests up in the hills. So all in all, an experience I'm very thankful for, and hopefully that impacted others as well. And I, of course, took some classes too. You got to do that to get a degree. So this included key skills like GIS, which I used to make maps. This is just an example, hopefully one that won't be needed, but I thought it was a good representative sample. And other things like quantitative methods, which I used the skills I gained in that class to produce these graphs. So hopefully you agree with me in thinking it worked, but I guess you have to make that determination for yourself. So with that being said, there were several key outcomes that I think arose from this work. So first off, helped to maintain healthy populations of these highly endangered Hawaiian birds. Of course, as I mentioned, the ʻalalā being foremost among them, but including the others as well. Generated this tool to help managers make informed decisions to add an additional tool into the box that was intended to be very accessible and flexible so that it could help represent this data in a nice visual format that would be useful for generating plans for the future. And also, I personally am very thankful to say I gained invaluable experience in a wide variety of skills. So as I mentioned, these key programs like GIS and different statistical softwares, learning how to code to produce this tool, and definitely had some long nights making that, but I feel like I learned quite a bit. So that I think was a success, and hopefully I was able to produce useful results for years to come. So what else, given the opportunity, would I like to see happen in the future with this tool and with the ʻalalā? Well, behavioral metrics of fitness are not captured in FLIC. And this is key. As I mentioned earlier, these birds are very intelligent. We want them to mate. So sometimes, whether they do or not is reliant on, for lack of a better word, their compatibility. Just like in humans, because they're very intelligent, some of them are more compatible and some of them aren't. So including these behavioral metrics of fitness or their ability to breed would be a welcome addition that I think would make the tool more robust, especially leaning into our ability to adjust the representation of each lineage. I think this could be a very useful component to add moving on. And additionally, as I mentioned, there have been ongoing efforts to reintroduce the ʻalalā. And we've noted through those efforts that survival in the wild requires a diverse variety of skills, and not all of which are very easy to cultivate in captivity. So for instance, one of the projects has been to try and induce a fear of their natural predator, the Hawaiian hawk, also known as the ʻio, in captivity. And this involves flying a rig with a taxidermied ʻio over the enclosure and playing appropriate alarm calls in order to have the ʻalalā associate that hawk with a predator that they should watch out for. So of course, if you grow up in an aviary and you never have to worry about getting eaten, you're going to have to learn somehow that those things flying around in the sky will come down and try and eat you. So this is one way we can address these differences between the captive and the wild environment. But I think incorporating these elements as well will be crucial to the long-term success of the program. So I think that just like including this element of compatibility could help make the tool more robust and factor in more accurately to these real life scenarios as we aim to reintroduce the species over time. Okay, well, with that, I would like to once again give my sincere thanks to Jolene Sutton, Bryce Masuda, Lisa Canale, Amy Kuhar, Allison Flanagan, and the rest of the just wonderful people that helped me along my journey. I don't, again, have space to give a shout out to everybody, but I would definitely like to give a big one to my friends and family who were supportive along this entire journey, and without whom I certainly couldn't see myself having arrived at this moment. So thank you very much. Appreciate your support. Also, of course, many thanks to the organizations and partners that helped me along the way, Hilo, KBCC, San Diego Zoo Global, and many others that I have not had enough space to name here. So thank you very much for your time and attention in this somewhat special edition of my presentation, and I will be happy to take any questions. Thank you. I can't see you, so hopefully everybody's good. All right, so I guess with that, maybe we'll open it on up to questions.
Yeah, please feel free to just unmute yourself and ask a question.
Great, yeah, and we have a little bit of extra time. If anyone has a question, don't be afraid to ask it, please.
Well, I have a question.
Yes.
You know those lines that you had that were all different colors?
Mm-hmm.
Okay.
Mm-hmm.
What did you measure to get those lines exactly? I mean,
so again, this is a simulated representation, but each of these lines represents the founder representation of dating back to one of those 9 original founders. So each of those lines represents a different lineage.
Right. But what did you measure to decide whether or not the lines went up or
so
down?
This is calculated from a technique which is a little bit difficult to explain. It's based on an additive genetic matrix of relatedness. So basically I wrote a program in language R that would consolidate the records taken from the extensive pedigree of the ʻalalā to be able to generate an additive matrix. And then using that step, you can then use the code to basically represent it graphically this way.
Oh, so is this DNA stuff? I mean, is this like genetic things that you measured to see how much variability there was or something, or?
In essence, this is based on the detailed pedigree that's kept of these birds. So it's not actually incorporating elements of DNA analysis into creating this graph, but it does underlie the need to represent each lineage separately, because it's assumed that they each have different DNA belonging to each lineage.
Right.
So we want to conserve them so we don't lose the allelic diversity, which is one way of measuring how different those DNA sequences are from each other.
Yeah. Fascinating. Okay, thanks.
Of course.
Jeremy.
Hi there, Leonard. Good to see you.
Yeah, a couple of, this is very interesting approach
Thank
to this whole problem. But one of the things that I was curious about, and how, if your model takes this into account, that if a lineage becomes overrepresentation, presumably it's more fit or better adapted or has more good alleles than populations that are gradually dying out. So because the ones that are dying out presumably have more subvital or perhaps even lethal.
Right. Yes,
So,
that's a very good question.
Okay.
Sorry,
so,
go ahead. If you have, did you have something else to add?
So how are, in these captive programs, how are these processes balanced?
Yes, that is a great question. Thank you. So as I mentioned, this tool is intended as one of many that managers use to keep these populations moving in the direction that they see is best for the species and its eventual goal of reintroduction into the wild. So given that currently all members of the population are in captivity, the effect of fitness in the wild and the effect of fitness in captivity are of course different, right? So as you mentioned, these lineages may be becoming overrepresented because they are more fit. That's the way we would see it in the wild traditionally. In this case, I can't say that I can speak to whether that's fully in line with what's going on in captivity, given that some of these birds, as I mentioned, are more, some of these birds simply might be more compatible or good at finding compatible mates. They might have better social skills, I guess, for lack of a better word. And this may be causing them to have more offspring and become disproportionately overrepresented. Now, that's definitely just my take on it. I think that that question goes beyond the scope of what I did, but I think it is a very good one. And of course, as I mentioned, adding these elements in would make this model much more robust. So I think moving forward, the more in line we can make the goals to keep for the wild population that may exist in the future and the goals for the captive breeding program line up, I think that would be very beneficial. Does that answer your question?
Yes. I have
Okay,
another
great.
one. Else wants to.
Good, good. Does anyone else have another question?
Jeremy, I have a question.
Oh, yes. Hi, John. Good to see you.
Hi, how you doing, Jeremy?
Doing well, thanks.
What are the main mechanisms you have for controlling the breeding? Are they mainly just isolating those birds that are overrepresented and then pairing the birds you're trying to get together? Are there any other mechanisms?
So that is also a very good question that I have some experience to be able to answer, but not give the full picture that you would get from one of the higher up managers. I'll do my best though. Essentially, there are, there's many different aviaries that birds are kept in. And there we go, thanks. And the birds are paired up with different mates to see what their level of compatibility is. Sometimes, of course, these decisions may continge on the compatibility is always being a factor, but they may also continge on thinking about the future of the population. So as I've mentioned, this is in addition to what's already going on, the attempts to minimize inbreeding play a large role in which ʻalalā are attempted to be paired up, because that is a mission critical goal, so to speak, is keeping inbreeding to a minimum, because combined with other best practices, that is considered to be one of the best ways to move forward, increasing, preventing the loss of survivorship that you would see with increased inbreeding and that loss of heterozygosity. So there are many different factors, but yes, isolating them in different aviaries basically is like the physical barrier that represents can be used to kind of control which ones they have access to mate.
Is there any artificial insemination ever done with these birds?
There is stored genetic material from some of them. And that might be a good question. I'm not off the top of my head sure whether I can discuss that, and also whether my mastery of how that program or how those efforts might be going is enough So sorry, I'm gonna have to leave you on that one for now. But since you
Thank
do
you.
live next to me, you'll get your chance to ask me again.
Look forward to it.
Okay, sounds good. Does anyone else have a question they would like to ask? I'll go back.
Yes, I have a question, Jeremy.
Oh, good.
The
I'm
idea is
glad to see you, Andres.
Good to see you. We're here, all the family rooting for you.
Oh, thank you very much.
The idea is that these captive birds, you let them out at some point in time to populate the you know, in
the landscape.
world.
Yeah, so there have been reintroduction efforts that have been going on since 2016. There have been some difficulties with the native predators, the EO that I mentioned earlier. I'm not sure if you caught that, but basically the Hawaiian hawk is one of their native predators. And of course, being evolved to hunt them, it's very good at its job, which represents a difficulty for us trying to keep them alive. So at the moment, all of the population is in captivity, but there's always the long-term goal of preparing them to establish in the wild again. So that is the goal of the program for not only the ʻalalā, of course, maintaining healthy wild populations for the other birds that still do exist in the wild is also very critical. Thank you for your question.
Excellent, thank you.
Thank you. Anyone
Hey,
else?
Jeremy.
Yes. Hey, how's it going?
Good. Sure. We got an echo here. Excuse us. We've got two
No worries.
devices
I can still
on.
hear you.
Okay. So, question is: Are there species that are similar enough that there could be interspecies mating, or are they exclusive?
That is a good question. Thank you. As for the ʻalalā, it's actually very unique. That's part of why I think it's so enthralling as a study species in many ways. It's a corvid in the Hawaiian Islands, which is, you know, very rare. The other species that are found on the islands don't really share, as far as I know, any, there's no potential for hybridization. I don't think that's ever been recorded to my knowledge. So that is not an element we'd have to worry about. And certainly in captivity, of course, being controlled carefully, having access to them and all that, the matings are one of the most controllable elements in most cases. That does, of course, as I mentioned, take into account the long-term goals, their compatibility and such things, but you can still have very good control over their access to each other. As for the other species that KBCC manages, I don't think that there's much going on in the way of wild interbreeding that's affecting the gene pools, but that is definitely beyond the scope of what I can speak to well. Does that answer your question? I hope so. Okay, good, thanks.
Jeremy, I have another question if I could.
Yes, of course, we have some time. So.
You know, where we're watching from, it's 4:43, which means it's almost dinner time, and looking at all those plates of food for the for the birds, it was intriguing. What were the ingredients that you were
yeah, so let me go back to that for a minute so we can talk about it. So there's a variety of different components. I did neglect to mention that earlier, so thank you for answering or for asking that question. So you can see in this picture, you might have to move your zoom window around a little bit, but you can see in these trays, we have some apple, we have some melon, some papaya, frozen peas and carrots. We have essentially what I would call bird kibble, basically like dry food, and then things even including live worms and cooked egg, and sometimes in some cases even frozen mice. So there's kind of a tailored diet for these birds, depending on their needs, sometimes individualized, but definitely always to the species level and the amounts. And each of these diets has been formulated by the excellent veterinarians working for San Diego Zoo Global. So they, you know, meet the nutrition standards for a healthy ʻalalā, so to speak.
Thank you very much.
Yes,
Good explanation.
yes, I'm glad. Thank you for reminding me to talk about that. Let's see. Does anyone else? It's great.
Hi, Jeremy.
Hey, how's it going? Good to see you.
Good, good. I just wanted to know, what is the survival rate of the eggs, for instance, that you have in captivity?
That's a good question. I can't put a number on it off the top of my head. Also, because like maybe I don't know if you caught this at the beginning, but basically because the information is not in the public domain for the species because they're so, they're very endangered and listed federally and on the state level. So I can't share all the information about them, but I can tell you that based on, this is kind of related, doesn't exactly answer your question, but based on a recent paper coming out just now in 2020 using regression tree analysis, it was noted that strong effects on survivorship are noted when you reach, when you get to birds that are somewhat inbred, an F value of about 0.1, you start to see a large drop off in their ability or their comparative survivorship with outbred birds. So in the captive environment, given how important and how focused management is on increasing individual survivorship and also minimizing inbreeding to support that plan, they are taking every possible step to make sure that that mortality rate is as low as possible. That being said, there are some challenges given that these ʻalalā, I guess it's kind of funny, but like you can imagine being a parent for the first time can be difficult. So there's challenges in the captive environment of being able to successfully rear these birds. Sometimes the parents can be their own worst enemy, quote unquote, knocking the egg out of the nest, even sometimes consuming the egg. So it's definitely not all of them, and it's more than we would like, because we would like to see all of them survive. And that this research and the ongoing work, of which I'm just a small part, is definitely focused on making sure as many of the population as many of the new offspring that come into the population as possible survive.
Thank you. And you started out with 200, is that right?
Currently, there's a less than 200 total individuals. That number has fluctuated throughout the years, but it is definitely a marked increase from the beginning of the program. And especially in recent years, refining the techniques to breed them in captivity has yielded some exciting improvements and results. So that's definitely good to see. Our understanding of how to be caretakers of these birds has improved, and that has definitely helped. And of course, the management strategies that I talked about and how my tool plays into them, all of those wind towards the same goal of making that happen.
Excellent. Thank you.
Thank you.
Hey, Jeremy.
Yes. Oh, yes, Leonard.
Hey, presumably the environmental factors that drove these birds to extinction have not changed, or perhaps gotten even worse.
Mm-hmm.
What do you think the long-range success of this program is going to be?
Yeah, so that's the million-dollar question, right?
Sure.
I think, and I'm sure Maya has told you, living in Hawaii, this is of course an issue that many people, especially in the conservation field, talk about frequently. There's certainly, just as in many places, competing interests between humans and animals. There always are. And Hawaii has a distinction in some ways of having ecosystems that evolved in isolation from a lot of the world and very far away from a lot of it. So this can alter, this can play a role basically in how that plays out that might be different to other places. In terms of the long-term success, I think this program is one of it's one of the leaders in terms of being able to successfully rear this many captive birds. And I think it's of course, you'd always want it to go better, but I think the progress that I've seen and I'm very thankful to have been a small part of, I think that's very heartening and good to see. I wouldn't be so bold as to presume I know what's going to happen in the future. We're of course all living through an unprecedented time of climate change and so forth. But I think with this much upswell of support from both the community and these different organizations, that can only be a good thing in giving these birds the best possible outlook on the future.
Well, what are the environmental factors? For example,
yes.
I'm familiar with the Plasmodium,
Mm-hmm,
and mosquito cycle. that I don't know if the ʻalalā is susceptible to.
Oh, that's definitely a good question. I know, of course, you'd want to know about the mosquitoes, huh? So that's good. The avian malaria has a differential effect on the different species, the different endangered ones that we've been monitoring the most. Some birds, such as the eevee, one of the birds I mentioned earlier, the bright red, it's very susceptible. It's thought that one mosquito bite is enough to transmit a deadly amount of pathogen and have for them to be susceptible and die to avian malaria, or of course weaken them and cause other factors to stress them more and collectively add to the stress load that can weaken them and cause them to die. The ʻalalā, given that it's been in captivity for such a great period of time, it's not as well known in my not as well versed opinion as many I'm sure you could talk to, but that is of course something that's consistently being thought of and on the table. So years ago, when we were discussing this project, one of the potential avenues of research that we also thought about was to try and associate different segments of genetic material, DNA, genes, alleles, with their ability to successfully resist or at least put up a better fight against avian malaria. So that's definitely part of the ongoing discussion, is being able to look at these factors, determine which birds may be potentially better suited for survival against them, and then ideally through this kind of same method, have those alleles become well represented in the population that's going to go out to the wild.
So you're genetically engineering a super bird.
I definitely would not say that, but I think that is you never know what's going to happen in the future, I guess. Great. Well, thanks, Leonard. I think, if I'm not mistaken, that might be the time we have. But Lisa, if you want to, how are you feeling? How's the recording?
The recording's doing great.
Okay, good.
We are coming up on time. If maybe there's a final question, we could open it up to that. Or if something came to you, Jeremy, and you'd like to share it as your final comment.
Well, I will ask first, I guess, if anyone has a final quick question for me.
Jeremy, I have a quick question.
Sure.
Maybe
I'm not sure. Sorry. Let's see. Yes. Thank you.
a couple of times you mentioned that the birds they're in captivity, and you mentioned San Diego Zoo. Are there some in San Diego Zoo, or are they just in Hawaii? I was a little confused.
At the moment, as last I've been informed, I obviously, because of this whole set of circumstances, I'm not there to see them as much as I once was, but they're currently all being kept in captivity either at KBCC or MBCC. There were plans to have essentially an exhibit at the local Panaewa Zoo in Hawaii to be able to have a great outreach opportunity. I'm not sure how those are going. I haven't followed up on that given the current climate of events. But of course, any time that you can expose people to how this amazing species and the unique aspects, that is a good opportunity to start a dialogue and start a discussion and generate support. So I would certainly hope, I'm obviously not in charge, but I would certainly hope that continues to be an important element as well. And I'm very certain the people in charge, they know what they're doing, so they're making it happen too.
Yeah. Good. Okay.
Great. Thank you for your quick question. I would just like to end with a heartfelt thanks for everyone who came to see me at this talk. I really appreciate your support. I hope you learned a little bit about this exciting organism and about how my work played a role in doing a small part in conserving it. So I would encourage you, if you have further questions, definitely feel free to email me. My email address there is written at the bottom. It's my last name, Schrader, and the first three letters of my first name, JER@gmail.com. So if you would like to follow up with me afterward, please feel free. Be happy to talk about it. If you can't tell already, probably like talking about this a little too much, so I might actually give you more information that you want to know.
Well, thank you very much, and thank you all for coming today. And In a couple weeks, we'll have this on the TCBES YouTube channel. So if you want to watch it again, it'll be there for your viewing. And so have a lovely afternoon, evening, everyone.
All right.
Thank you very much, everybody. Thank you.
Congrats.
Thank you. Appreciate the support very much.